Okay, our next speaker in this morning's session is Ashley Kiemen from Johns Hopkins University. And for the people attending on Zoom, please, we're monitoring the Slack channel if you want to put questions in there. People in the room can use the Slack channel too.
I don't know how to use Mac. Just a second. Okay, thank you all. My name is Ashley Kiemen. I'm a faculty at Johns Hopkins, and today I'll be speaking on behalf of Jae-yoon Song, who's a PhD student on the team who had to be out of town today. Jae-yoon's thesis is on a novel classification of phenotypes of Panin lesions that can be identified along multiple length scales using a tissue mapping technique developed at Hopkins called CODA. To give a quick motivation of why we study Panin lesions, PDAC is one of the deadliest malignancies, both because it develops slowly and with symptoms that can be easily confused with something inane like food poisoning, and because at the time of diagnosis, PDAC has often developed distant metastases, meaning we have few clinical options. So what we've kind of decided as a team is that to best fight this deadly disease, we should move back in time and study its initiation. And PDAC most commonly develops out of Panin, which is a non-invasive neoplastic lesion in the pancreatic ducts. It's come out lately that people contain an unexpected amount of Panin, especially as we age. All of us will develop these Panins, which is a bit confusing given how few people develop PDAC. And despite all of this, we don't know a lot about these lesions. So to show you what they look like, Panin is most commonly seen in histological slides. So this is an H&E image, which is a common means of studying tissue anatomy in the clinic. Panin is too small to see in diagnostic imaging like CT and EUS. So up until not too long ago, it was almost only studied in H&E. So because of this, we only had a sense of what it looks like in 2 dimensions. And that didn't allow us to study things like connectivity, genuine incidence of these lesions in the human body, and how they might connect with additional anatomical tissues. So actually, Panin was what motivated the initial development of CODA, which is a tissue mapping technique that we now use extensively at Hopkins to study many diseases. And what CODA does is it takes advantage of histology and the fact that with H&E slides, we can see single cell level, we can see a lot of anatomical components, and with digital pathology, we can map H&E. So what CODA does is instead of taking a single cut to study anatomy in 2D, we'll keep cutting down a block and get tens to thousands of slides and then apply a sequence of computational techniques to align all those slides back into a digital volume, to label the anatomy with AI, and to then build back these quantifiable and visualizable maps. So just to give a quick conceptual view of what it does, on the left-hand side, you see a sample set of tissue analyzed by CODA. So this is about 1,000 slides that you see jiggling because it's physically cut tissue. So unlike a lot of the imaging techniques we've seen this week, CODA uses physical sectioning and scanning to build back digital volumes. So step 1 is just let's take all these independent tissue slices and line them back up in space. And then step 2, I like to call it quantifying the pathologist. So we use a segmentation-based technique to use a set of manual annotations that can be customized to any tissue sample that we analyze, and allows us to automatically detect the types of tissue that a pathologist could see in H&E. So what's so special about that is no single antibody could be used to stain all of a Panin cell without also staining something else. So if you stain epithelium, it's going to also stain the healthy ducts. If you stain With subtypes of mucin, as we'll see, it only stains some pannins. So we need a technique that utilizes H&E and detection in H&E to study these lesions. So the initial question was, using CODA, can we go beyond what's been done in the past? Can we look at the connectivity of pannin, and can we finally see what they look like beyond 2 dimensions? So in this video, you see the AI is automatically following the neoplastic cells along a sequence of slides. And if we stack those all back up and visualize them, we can see now that pannin is not just a little disc, but it's actually an octopus. So it's developing in this ductal system. So as you might expect, it comes out with all these connected tubes. Scaling up, what we did was we applied this technique to a sequence of thick slabs of tissue, each one of which is about the size of Monopoly dice. And unexpectedly, I can think back to meetings with pathologists a long time ago that we came into the meeting and said, oh, this sample has 52 pannin. And they said, no, it doesn't. Just because when you look in 2D, you might see a lot of independent little lesions in a duct, but you might expect them all to connect. And what we found was you might have a couple big connected pannins, but you also have a ton of small ones. So scaling that up into many samples, in a study that just came out a couple weeks ago, we showed that individuals have, again, just a shocking amount of these pannins, up to thousands as we age, and that we expect most of us will get these. So this gave us some new questions. If all of us will get pannin lesions, but so few of us will develop PDAC, about 1 in 60 of us, what's unique about some pannins that allows them to continue to develop and become invasive? Can we identify how pannin initiate and what might distinguish some pannin distinctly? So that's what Jae-yoon has been following up on as a PhD thesis. And she's been following up on this initial finding that we had, that if we take a top-down view of some of these samples, what you see in this image is the ductal system in blue and then some pannin lesions kind of laid on top of it. And this image is a good example of what we thought pannin would look like, is they would look like tubes. And we did find that a lot of them look ductal because they develop in ducts, it makes sense, but that some of them look dilated, like this one in the middle, like a blown up balloon. And that some of them look solid and clumpy, like the lobules of acini. So what Jae-yoon has done is she noted that these phenotypes, as we call them, can be initially seen on a global scale. So on the scale of a whole block of tissue that can contain billions of cells. But taking advantage of one of the benefits of CODA, that we can always go back to the 2-dimensional histology to single cell level, she asked, can we identify this global phenomenon in 2 dimensions in H&E? So initially, she just collected some samples of what each of these look like, and you might be able to see immediately that we have distinct phenomena going on. All the way on the left, you see these like wavy papillated epithelium with swollen nuclei and maybe even distinct cytoplasm. In the middle, you see a nice, neat line of cells. I like to think of them as almost well-behaved cuboidal nuclei. And then on this side, you see pale cytoplasm, and the nucleus is squeezed along the outside edge of the cell. So using this, she spent a lot of time developing this novel classification scheme that takes advantage of changes she notices at the nucleus level, the cytoplasm level, the shape of the cell, and the change in the size of the cell between the basal and the apical face, which looks a bit like this. As soon as she developed this schema, she also noted that the same classification scheme actually exists in healthy anatomy in the stomach. The same shape of the cells, and even this shape has been linked to antibody signaling. So pannin is defined as containing mucin, but multiple types of mucin exist. And she found, just like in the stomach, that MUC5AC and MUC6 can be used to distinguish these phenotypes. So we have the ductal in blue and the solid the ductal in pink and the solid in blue, and they can be distinguished by 2 mucin stains. So now we know that this high-scale global phenotype can be identified at the single cell level in H&E and can be validated using antibody labeling. So what she's been spending time on now is, now that we've qualitatively defined these, developed a systematic classification scheme, can we do the same thing with AI? And can we go back to that huge data set of samples that we've collected, that we've mapped thousands of pannin lesions, and quantify the composition, the incidence of these phenotypes in those samples. So can we systematically map this classification scheme? So she's taken advantage of the segmentation that we do with CODA, and she's added a little twist to it. So because we have existing classification of the anatomical subtypes of these samples, all she has to do is subclassify the pannin into these phenotypes she's identified. So what she's been doing is annotating a subset of slides, building a model, and then using the model to gain new annotations. So annotate one slide, classify a new slide, edit those annotations, and plug it back into the model to quickly build a big data set that she needs to classify all these blocks efficiently. And this looks like a simple little table, but I think it took about 18 months of painstaking annotations and double checking and making edits. But she finally has high quality classification showing that we can distinguish these phenotypes in, I think the data set contains 50,000 H&E slides, and she's testing on about 75 of them. So this is well validated between many patient samples. To show an example of what that automatic labeling looks like, in this slide, you see a lot of pannin, and she's labeling all the distinct phenotypes. So what we saw immediately, especially if we zoom back out to the global scale, is that now that we've applied AI at the single cell level, it does map back to that global anatomy that we saw at the beginning. So the ductal phenotype we can see is labeled by that pink label, the MUC5AC positive. The yellow shows up in the dilated locations of the ducts, and the blue solid phenotype is kind of budding off the pannin. Oh, I was supposed to be clicking. So they show up when we classify at the single cell scale back to the global scale. And she just got to this point about a week ago, so we have some conclusions and next steps on this study. So we found in the past that pannin exist in a shocking abundance in human tissues. And when we can subtype these pannin into these distinct phenotypes, they can be identified on the global scale within H&E and using antibodies, validating that something biological is going on. What we have next to do with this classification scheme that Jae-yoon has developed is to quantitatively map these phenotypes in a huge data set containing thousands of pannins. And we have questions about how the phenotypes might play into the initiation of pannins. So what is the phenotype makeup of the biggest pannins and the smallest pannins? And finally, to add in DNA sequencing and Visium to see what else might distinguish these anatomical subtypes. So with that, thank you, and I'm happy to take any questions, and especially thank you to Jae-yoon.
Thank you, Ashley. Questions? Said I'd check the Slack, so I'll do that.
Have so many hundreds of these.
Mm-hmm.
Where is the cancer coming from?
Yeah, that's kind of the question we have, is it seems like most of them might just be a benign thing that happens as we age. And I think the same questions have been happening in IPMN, which is the second lesion that can develop into PDAX. Because IPMNs can be seen in CT imaging, people had these questions about a decade ago of suddenly they've been scanning and detecting with AI these IPMN cysts, and initially thought, wow, we have so many IPMNs, let's take them out. And then quickly it became, wait, we all have IPMNs, we should not be taking them out. So yeah, I think it gives us a unique way to ask which ones might be developing into PDAC.
Thanks. So these are much larger than you would think they would be. So
Yeah.
Why don't we see them in any sort of clinical imaging modality?
Yeah, so they can get quite big. I mean, this looks small to you, but as cysts go, it's huge. I think even though it's big, because it's thin, I think it's still not visible in things like CT. But what we've begun to do is we know that while pannin is confined to the ductal system, when it clogs the ducts, it's associated with things like fatty deposition, collagen deposition, and loss in the acini, the lobules. And that can be seen at the bulk level. So we've thought about, if we can't see the pannin, can we see something it's associated with?
Thank you. Great presentation. So I know you said that you're going to do spatial transcriptomics and sequencing, but I was wondering if you had any data in terms of the mutational status, and if what you're looking at is a field effect of sorts and some sort of global premalignant transformation that we're witnessing, or if these are distinct lesions that develop with a different mutational profile. Thank you.
Yeah, so that's a good question. In the study that just came out about a week ago, we mapped pannin incidence and then used DNA sequencing to look at somatic mutations between some of the biggest pannin, and found that they tend to be independent. So they tend to have unique clonal events. And that some of the biggest ones, like this spinning video is the biggest pannin, might be composed of multiple clones that meet in the ductal system.
Train a machine learning algorithm to recognize the mutational profile of distinct pannins to the way they look on H&E. And perhaps at some point with thin cutting through the pancreas, maybe there is even a clinical imaging that can be utilized to tie into the appearance of histology and a step forward to the molecular characterization of these pannins, which would have prognostic implications.
Yeah, we've actually talked about doing that. I think with the existing genetic data we have, it's not quite enough to build a good model, but it's definitely something that's been kind of stewing a while. I would love to do it.
Okay, let's thank Ashley again.